Hello students, today let's discuss about causes of mutation and types of mutagens. In introduction, the development and function of an organism is in large part controlled by genes. Mutations can lead to changes in the structure of an encoded protein or to a decrease or complete loss in the expression. Because of a change in the DNA sequence affects all copies of the encoded proteins, mutations can be particularly damaging to a cell or organism. In contrast, any alteration in the sequence of RNA or protein molecules that occur during their synthesis are less serious because many copies of each RNA and protein are synthesized. Geneticists often distinguish between the genotype and phenotype of an organism. Strictly speaking, the entire set of genes carried by an individual is its genotype, whereas the function and physical appearance of an individual is referred to as its phenotype. However, the two terms commonly are used in a more restricted sense. Genotype usually denotes whether an individual carries mutation in a single gene or a small number of genes, and phenotypes denotes the physical and functional consequences of the genotype. Dominant and recessive mutation. A fundamental genetic difference between organisms is whether their cells carry a single set of chromosomes or two copies of each chromosome. The former are referred to as a haploid, the latter as diploid. Many simple unicellular organisms are haploid, whereas complex multicellular organisms, as for example fruit flies, mice, humans, are diploid. Different forms of a gene, as for example normal and mutant, are referred to as alleles. Since diploid organisms carry two copies of each gene, they may carry identical alleles, that is, be homozygous for a gene or carry different alleles, that is, be heterozygous for a gene. A recessive mutation is one in which both alleles must be mutated in order for the mutant phenotype to be observed. That is, the individual must be homozygous for the mutant allele to show the mutant phenotype. In contrast, the phenotypic consequences of a dominant mutation are observed in a heterozygous individual carrying one mutant and one normal allele. Recessive mutations inactivate the affected gene and lead to a loss of function. For instance, recessive mutations may remove part of or all the genes from the chromosome, disrupt expression of the genes, or alter the structure of the encoded protein, thereby altering its function. Conversely, dominant mutations often lead to a gain of function. For example, dominant mutations may increase the activity of a given gene product, confer a new activity on the gene product, or lead to its inappropriate spatial and temporal expression. Dominant mutations, however, may be associated with a loss of function. In some cases, two copies of a gene are required for normal functioning, so that removing a single copy leads to a mutant phenotype. Such genes are referred to as haploinsufficient. In other cases, mutations in one allele may lead to a structural change in the protein that interferes with the function of the wild-type protein encoded by the other allele. These are referred to as dominant negative mutations. Now let us discuss about gene mutations and chromosomal mutation. In gene mutation is a small-scale alteration of the genetic material of an organism, which primarily is a change in the nucleotide sequence of a particular gene. These changes are of two types based on the way this takes place. Point mutations and frame shift mutations are the two main types, but the frame shift mutations takes place as either deletion or insertions. When a nucleotide of a particular gene is changed, the transcribed mRNA and the subsequent codons and synthesized amino acids are altered. However, there are few other subtypes of point mutations known as transition, transversion, silent, missense, and nonsense. The frame shift mutations, also known as framing error, takes place after the transcription of protein synthesis as an extra nucleotide is attached or insertion to the dismantled DNA strand. Additionally, there could be a chance of losing a nucleotide from the unwound DNA strand after the transcription. Gene mutation may lead to alter the number of the structure of the entire chromosome, which could lead to chromosomal mutations. However, gene mutations are small-scale alterations in the nucleotide sequence, and these are sometimes corrected through gene regulatory mechanisms, but sometimes not. Some of the problems that are evident of a gene mutation cause are the sickle cell anemia and lactose intolerance. In chromosomal mutation, chromosome mutation is a large-scale alteration of the chromosome of an organism where either the number or the structure of the chromosome is changed. There are three main types of chromosomal mutations known as duplication, inversion, and deletions. When a particular part of the DNA strand is duplicated or doubled, the number of genes in a chromosome will increase, which causes both structural and numerical alteration to the chromosome. Sometimes a portion containing genes in the DNA strand gets removed and joined inversely to the original position, which results as an inversion. Inversions do not cause the number of changes, but different interactions may be resulted as the gene order has been changed, hence the phenotypes becomes different. Deletions may take place due to exposure on radiation, high heat, or viruses. Usually, deletions are result of external causes, and the affected area of the chromosomes determines the extent of change or the damage. All these chromosomal mutations seriously affect the structure and the number of chromosomes in an organism, and the eventual protein synthesis and gene expression will be altered. Prader-Willi syndrome and Cry-du-Chat syndrome are some of the examples of chromosome mutations taken place through deletions. Mutations can be classified as induced or spontaneous mutations. Induced mutations are those that are induced purposefully with exposure to mutagens, whereas the spontaneous mutations are those that arise spontaneously without intentional exposure to the mutagens. Spontaneous mutations are the source of natural genetic variation observed in a population and have played a vital role in evolution. Now let us discuss about types of mutagens. Mutagens of induced mutation can be classified as biological, chemical, and physical mutagens. In chemical mutagen is a mutation agent which is in the form of chemical substances. It can mimic nitrogen base in normal DNA, but they cannot couple during DNA replication. Moreover, chemical mutagen has an ability to insert between nitrogen bases and disturb the DNA replication. In base substitution, some chemical compounds are similar to the nitrogenous bases in the DNA and can be incorporated in substitution of the normal base. This type of compounds are called base analogues. Unlike the normal bases present in DNA, the base analogues have a different pattern of pairing and can induce mutation by allowing insertion of incorrect nucleotides during DNA replication in a process called tautomeric shift. As for example, 5-bromouracil, which is an analog of thymine, promotes base pairing with guanine instead of adenine. In base alteration, some mutagens are not incorporated into the DNA but instead alter a base, causing specific mispairing. Certain alkylating agents such as ethyl methanesulfonate also known as EMS, and the widely used nitrosoguanidine NG are examples of such mutagens. In intercalating agents, the intercalating agents are another important class of DNA modifiers. This group of compounds includes proflavin, acridin orange, and a class of chemicals termed ICR compounds. These agents are flat planar molecules that mimic base pair and are able to slip themselves in between the stacked nitrogen bases at the core of the DNA double helix. In this intercalated position, an agent can cause single nucleotide pair insertion or deletion. Intercalating agents may also stack between bases in a single-stranded DNA. In so doing, they may stabilize bases that are looped out during frame shift formation. Physical mutagens. Physical mutagen is a mutation agent which is in the form of physical substances such as short wave, ultraviolet and radiation ray such as alpha, beta and gamma. For example, radiation from radioactive substances such as uranium, radium and cobalt. X-ray and cosmic ray can damage DNA. Damaged DNA molecule cannot undergo usual process of replication and transcription. A large number of mutagens damage one or more bases, so no specific base pairing is possible. The result is a replication block because DNA synthesis will not proceed past a base that cannot specify the complementary partner for hydrogen bonding. In bacterial cells, such replication block can be bypassed by inserting non-specific bases. The process requires the activation of a special system, the SOS system. SOS induction is a last resort and used as an emergency response, allowing the cell to treat death for a certain level of mutagenesis. However, the repair mechanism is error-prone and can lead to mutation. Let us discuss about biological mutagens. Biological mutagen is a mutation agent in the form of virus or bacteria. Some of the examples of biological mutagens are rubella virus, cytomegalovirus, and hepatitis B virus. Transposons can also be considered as a biological mutagen. We will discuss about spontaneous mutation now. In spontaneous mutation arises from error in DNA replication, spontaneous lesions, as well as insertion of transposable elements. Spontaneous mutations are very rare, making it difficult to determine the underlying mechanism. Some of the factors that can lead to spontaneous mutations are error in DNA replication. Mispairing in the course of replication is a source of spontaneous base substitution. Most mispairing mutations are transitions. This is likely to be because an A, C or G T mispair does not distort the DNA double helix as much as A to G or C to T base pairs do. However, transversions also can occur through mispairing. Replication error arises due to low fidelity of DNA polymerase or an error in the proofreading activity. Spontaneous lesions. Naturally occurring damage to the DNA called spontaneous lesions also can generate mutations. Two of the most frequent spontaneous lesions are depurination and deamination, leading to change in nucleotides. In conclusion, mutation is a process that is essential as well as deleterious. Some of the examples of diseases arising from point mutations are cystic fibrosis, sickle cell anemia, Tay-Sachs disease, phenylketonuria, and color blindness, among many others. Mutagens are classified as chemical, biological, and physical. The process of mutation is an essential process of evolution but has undesirable effect too, as mutation can cause cancer and inheritable as well as non-inheritable genetic diseases that has no cure. Considering the extensive damage that mutations can result, precautions should be taken while handling possible agents of mutation.